Welcome everyone to yet another episode of Cellular Conversations. My name is Marta Gabrielska. I'm from Millennium Science, responsible for 10x Genomics. And today I have with me Jeff McDermott from 10x Genomics, he's Science and Technology Advisor, and our fantastic speaker, Somi Kordafshari from Jane Visvader Lab at WEHI. Last time, if you were attending, we were talking about the FLEX assay, and today we'll be jumping into a multiome assay. And you must know that multiome is not maybe as simple as some people might think. It's very good assay. It's combined from two parts, gene expression and ATAC-seq at single cell resolution, which makes it sometimes challenging and some people might struggle with it. However, Somi, for some unknown reason, and the reason is probably being born with the pipette in her hands, which I repeated last time, she just basically make it really work fantastically in her hands. So it is truly rare that to see someone who can make every single 10x assay work flawlessly and consistently generate high quality data. So with that little very simple introduction, I would like Somi to present to you. Somi, the audience is yours.
Oh, sorry.
Go back.
Okay.
Yeah.
Okay. Thank you so much, Marta, for the very kind introduction. And hello, everyone. I'd like to begin today by acknowledging the traditional custodians of the land on which we gather meet today and pay my respects to their elders past and present. Well, as some of you might already remember from my last webinar, I've been running pretty much all of 10x assays in our lab breast cancer here at WEHI across human, mouse, and diverse tissue with over 300 samples to date using assays listed here. So last time I walked you through the amazing FLEX assay that I've been using on our rare breast cancer subtypes, and I showed you how fascinating it is that we can get such a high quality single cell resolution data from such degraded FFPE. And hopefully next time I can share some of our very exciting high resolution xenium data that I have got from our same breast cancer samples that I processed with FLEX. Speaking of which, before diving into our today's focus, here's a bit of breaking news that I'd like to share with you about the new version of FLEX, which is GEM-X FLEX, which was launched recently. Since my last webinar, I've been using this new version of kit as for the first time as part of our ongoing rare breast cancer project. So within the past few weeks, I have already processed 12 FFPE, and as one of the early users of this kit, I can confidently say it's just amazing. I mean, I was thinking that if I could make a gallery out of my FLEX runs, this round of data would definitely be a showpiece. And here's why I love it. You can get 20,000 cells from each sample, which makes the high throughput FLEX profiling much more powerful and efficient. You need way lower input. It's just less than 50,000 to maximum 300K, which used to be 100K to 2 million. And now the recovery has improved from 60 to about 80, meaning using less nuclei, still you achieve high recovery. And on top of all, the undetectable multipellet rate is impressively low. It's just 8% per 20,000 cells. And here, showing some of my sequencing quality and performance metrics. So I got this many cells, 80,000 from 4, 50,000 from 3 samples. And the fact is, having this many cells makes the data set much more representative of the true tissue composition, rather than just a few snapshots by taking only a few thousands of cells, right? And then having this high UMI, 500, 600 millions that I have, meaning that I have captured way greater transcriptome complexity, which is especially important when you are profiling very heterogeneous tumors like our cases, where the cell types and states are still unknown, and you are the first person that need to accomplish this mission. And finally, having this high saturation gives me the confidence that I have effectively captured and decoded whatever was in those cells without missing any major signal. And then breaking these metrics into sample, you can appreciate a decent number of median gene and median UMI per sample, which is actually quite impressive, because don't forget that these are FFPE, not fresh sample, and these are nuclei, not whole cell. And then projecting these metrics on the UMAP, you can Yeah, so projecting these data on the UMAP, you can clearly see how clean it is with well-separated clusters capturing all expected cell types. And then another highlight I wanted to, that I missed in my last webinar is that we all know that capturing neutrophils are quite inherently difficult, right? Especially from fresh samples because they have lower RNA content, higher RNase activity, and shorter half-life. So basically, they easily get lost during handling. But I've been consistently getting distinct neutrophil cluster, as you can see here, here, and here, and which is thanks to high sensitivity of FLX, which is now much higher with GEM-X FLX. And the last point I want to address is related to a question that was raised in my last webinar about how FFPE fixation impacts downstream analysis. So as I mentioned, as a general rule, especially, ideally the tissue upon removal from patients should go to the formaldehyde, shouldn't be on the room temperature in OR, and then not more than 18 hours. So far, whatever I've got were from RMH and Peter Mac, and they all worked well without, for example, me needing doing DV200. However, recently I got 2 FFP from another hospital. Although I was able to get millions of nuclei, but I couldn't recover any meaningful data from these cells. So, and more interestingly, the corresponding xenium data from these samples also showed extremely low RNA content, which means whatever they are using as a fixation protocol, it's not compatible with RNA preservation. So that's why I really want to emphasize that FFPE fixation matters when it comes to studying RNA. And yeah, if I can get some information about how these fixation differ across pathology resources, definitely I will share them with you. So let's get back to our today's focus, multiome assay. Well, our team works on the very complex and heterogeneous breast tissue, looking across different life stages, hyperplastic and tumorigenic contexts. And the fact is, to really understand this complexity, we need to look at both transcriptional and epigenetic landscape. And that's where the 10x multiome has been like a game changer for us. Because with multiome, you have this luxury of capturing both gene expression and chromatin accessibility from the same nucleus, which is key for linking cellular states to the regulatory programs. So for today, I'm gonna show how well this multiome performs, even with challenging archival frozen samples. And I'll share some of my hands-on expertise just to assure you that you can establish and set up a robust workflow that would give you reproducible, high-quality data. But let's see, why do we study normal breast? Because for this project, I have been using multiome on normal sample. Well, let's start with a brief overview of its structure. The adult normal breast comprises an intricate epithelial ductal tree that ends to a cluster of acini called TDLU, the functional unit and where the majority of tumor arise. And in fact, the paracrine glandular function of breast mediated by these highly branched lobules that produce milk and then transported to this duct network. So this is a H&E from one of our patients underwent reduction mammoplasty that shows these lobules and ducts, and that these are embedded in a adipose-rich tissue, a dense vascular lymphatic vessel, and ECM-rich connective tissue. And basically, the signaling and crosstalk of this microenvironment and this epithelial tree influences both normal physiology of the breast and carcinogenesis. And this is a simple model of breast cancer pathogenesis, which is a result of proliferation of abnormal cell from duct or lobules that tend to a pre-invasive form DCIS. But once the tumor cell breaches the basement membrane and infiltrate into a stroma, then it is called invasive carcinoma. So now you can appreciate why studying the normal breast is so important, because it has implication not only for understanding breast initiation, but also how reproductive or hormonal factors such as menstruation, pregnancy, lactation influence molecular and cellular events in breast normal dynamic. But why do we use multiome? So let's briefly say what is epigenetic and why it's important. So epigenetic refers to the molecular mechanism that controls gene expression without changing the DNA sequence. This includes things like chromatin accessibility, histone modification, DNA methylation, acetylation, and essentially these are the factors that determine if genes are turned on or off in a cell, which shape the cellular identity and eventually how it would respond to the environment. So in the context of normal breast, to really understand the complex physiological influence on normal biology, we need to capture complexity of breast at multiple regulatory layers. And this is where 10x multiome comes in, which allows you to capture GEX and ATAC from the same cell. So here on the left, you can see the traditional approach. You split the sample, some goes to RNA, some goes to ATAC, but then you need to infer the link between these two contexts. But that's indirect, right? And you can miss cell-to-cell variation. For example, how would you know? I mean, each cell may have a unique chromatin state and expression profile. And when you split the sample, you are literally losing the ability to see which regulatory changes correspond to which transcriptomic changes. But look, on the right side, we have Multiome, where you can measure GEX and ATAC from the same cell. And here, you can directly link gene activity to regulatory elements without guessing. And that's the highlight. That's the point. There is no guess here. And let's be honest, people often say that multiome is too expensive. But if you ask me, you really need to look at the whole picture. Like many other things in our life, something is cost effective when you get far more than what you pay for, right? And with multiome, you truly do. You are capturing 2 layers of data, getting beautifully complementary insights from the same cell. So yeah, I mean, I would say that even it should have been more expensive compared to other 10x kits. So here's a brief overview of my workflow. We start with normal breast, dissociate into fragments, freeze to create archival sample. Then at the time of experiment, we thaw, make a single cell suspension, fax sort to get the total cell. Then either I straightforward use the cell for GEM-X to capture the whole cell RNA, or I extract nuclei for multiome. So before moving on, I want to give a big shout out that this multiome and RNA is definitely not just my effort. All the samples I received for nuclei isolation were expertly processed by my colleague Francois. And in fact, these 3 yellow steps, they were all done by him. And he's our in-house expert for prep and also establishing PDX models with truly magic hands. Believe me, if he can, I mean, he can get literally high quality cell from any kind of sample, like turning the stubborn tissue into gold. And I'm really grateful and lucky to work with him, because at the end of the day, the clean data doesn't start from me. It starts with his magic hand at his bench. So moving to the main focus of today's webinar, establishing a robust multiome workflow. So I'm going to keep it brief and highlight the most critical steps. So nuclei isolation, the foundation of high quality data. Once you get high quality nuclei, do the transposition carefully and gently, then generate the gem. Everything from that step onward becomes much smoother. Here I'm following this protocol, but I have also used this one, which is for the isolation of nuclei directly from tissue-derived cells. However, for this particular project, because our samples that met our selection criteria were available as frozen fragments in our sample archive that already undergone a lot of enzymatic processing, so I had to use this protocol. But please note that before going through the details that the guide already highlights the critical steps with clear attention symbols wherever needed. So it's a robust, super robust protocol. And if you just follow it, you will get high quality. But if not, you will end up to a suboptimal performance. Now I'm going to sharing some of my hands-on tips that would allow you to get reproducible data beyond just following the written protocol. Buffers. So always prepare fresh just before starting the prep. Nuclei lysis must be on ice using chilled reagent. Use only the reagents, believe me, use just the reagents and consumables recommended by the protocol. Actually, this is not the time to save money because you have already invested in this assay, right? So cutting corners here just and compromising your result. And every reagent in the protocol to me has a strong rationale, so just trust it and follow it. For example, the RNase inhibitor from Sigma might look a bit more expensive than alternatives, but it is specified for a reason. That said, you can still save this expensive enzyme. I mean, this is what I have been doing by preparing a smaller buffer volumes. I personally avoid processing too many samples at a time. Let's say, for example, I mean, I will explain why later, but let's say for 3 samples, I'm talking about lysis. Let's say for 3 samples, 100 microliter each, let's add 10% extra, you need maximum 400 microliter. So in which you can save this RNase inhibitor, which is expensive, right? But to pipette accurately small volumes for this lower volume of lysis buffer, I always dilute the concentrated stock of sodium chloride, tris, digitonin, and DTT, which allows me to prepare small volume without wasting too much RNase inhibitor or running into pipetting errors. And then, before going to isolation, and then for the resuspension buffer, for each sample, you would need maximum 80 or 100 microliter. So instead of making 1 ml, I would say if you make 400 or 500 microliter, it would be more than enough. So you don't need to make 1 ml. Here, again, you are saving reagent. So for the isolation, the protocol specifies 100K to 1 million, right? I personally, based on my experience and optimization, I prefer not to go to exceed beyond, let's say, 400 or 500,000 cells, because based on my experience, this seems to be an optimal number for efficient lysis. I reckon increasing the cell input too much could reduce lysis because the reagents may not fully diffuse. And I think lysis efficiency depends on the ratio of cells to reagent, not just the absolute number. So if you overload, some cells may not get fully lysed and give you lower nuclei yield. And then when it comes to lysis, you have to pipette it very extra gentle 10 times, but be fast. And keep the key here is that you need to, this is what I would do. So you need to keep the tip along the wall of the epi to minimize the shear stress on nuclei. And here in lysis, believe me, timing is super critical. You need to optimize it for your own sample, but for the optimization, you don't need to add expensive RNase inhibitor. So just test different duration until you achieve the best nuclei shape. In my case, I use 5 minutes. And for this, you need to stagger timing for multiple samples. Because let's say we are processing 3 samples, right? When you add lysis to the first tube, do 10 times pipetting. By the time you start sample 2, at least 1 minute has already passed from sample 1, and so on. So to avoid over lysis, I set separate timer for each sample rather than starting the timer after finishing the last one. This way, every sample gets the exact optimized lysis time, because to me, even a few seconds matter. So we want to avoid under or over lysis, because remember, for the attack part, we want to profile DNA in its native chromatin context, which reflect open to closed chromatin, right? So when you overlyse nuclei, they get ruptured, DNA leakage happen, the DNA is no longer protected, it becomes linear, free, it means more accessible for transposase, right? Then that enzyme starts to cut everywhere, and then you would generate a lot of random fragments that don't correspond to true open chromatin. And that's why overlysis leads to artificially high background and poor peak definition. And also, we don't want to end up with underlysis, because it results in cytoplasmic debris, which leads to poor transposition efficiency. If you couldn't see the pellet at the start of the experiment, don't panic and don't waste your time by thinking why someone else could get it. Don't compare yourself with anyone. Just transfer it to a 500 microliter tube. Although it's 500 microliter, but you can fit up to 800 microliter, because the smaller button makes the pellet more visible. But make sure that you have the appropriate tube adapter for centrifugation. And then when it comes to the final resuspension for the next step transposition, let's say I would aim for 10,000, right? So I always aim for concentration of 3,500 to max 4,000-ish, not too concentrated, because since the maximum input for transposition is only 5 microliter, you don't want to end up pipetting tiny volumes like 2 microliter, because smaller pipetting volumes increase the chance of error. So for the nuclei counting, first, I check quality under the microscope. This is the point. I use a very diluted sample for checking the quality, because this prevents clumping and allows you to inspect each nucleus individually. So you know that the intact nuclei look round and smooth, while the compromise here, they look ruffled, like indicating bellowing. So, and ideally, you shouldn't get a live cell. So this is my own prep, although I have taken this picture with my mobile, very low quality, but still you can see that they are very generally homogeneous, uniform in appearance, and I don't get any dead live cell. And if I see aggregates to this extent, I wouldn't be worried because you can break them up by pipetting. Another thing is that I personally never ever vortex my nuclei in Multiome. Remember, these nuclei have already experienced too much stress. Freeze, thawing, sorting, exposure to a strong detergent during lysis, and vortexing can damage them further. So I would say gentle handling is super critical. So just to break up these aggregates, I would do few gentle pipetting, and then for counting, I would use PI with automated counter. And yeah, I mean, a low percentage of live cell is accessible, but you don't want to have a mix of live and nuclei for several reasons. Because of main one is RNA bias. Live cells have cytoplasmic that will skew your RNA measurements. And then because of the ATAC efficiency, transposase tagmentation work best on nuclei, not whole cell. And then it would lower the efficiency of tagmentation, which in fact, down the track would reduce the ATAC quality and lead to uneven fragment size distribution or other QC metrics. And actually, this is my favorite slide. So after the nuclei prep, which I would say that most critical upstream step, and honestly, the one where most people struggle with and things most often go wrong, I want to share a few key checkpoints for downstream. And I can assure you, if you simply follow the basic but fundamental molecular handling principles, everything works beautifully because again, all of 10X kits are super robust. But the problem is that, which I have seen from many hands, from students even to senior postdoc, people often chase the fancy side, kits, platforms, algorithm, but literally they are overlooking the basics, and that's where the data quality is truly made or lost. That's why I decided to move away from the usual cliche that you would see in the workshop or webinar and take a step back and walk you through a few really basic but absolutely critical. And I personally can guarantee if you stick to these few things, really stick to them, everything else just falls into place naturally. So never rush an experiment or skip even the tiniest step. That a small thing is often what decides success or failure. I keep telling everyone, you are not the winner just because you finished first. The real winner is who produce clean and reproducible and high quality data, even if it takes longer. So to me, every step is like a link in a chain that if you miss one, the whole structure breaks, right? So this is my first rule or principle. Patience is your strongest reagent. It's not just about the speed, it's about the precision. And then for troubleshooting, always start simple. Never underestimate the power of the most basic, undeniable factors. I have seen this many times, again, from students, postdoc, that they tend to overthink the problems as if keeping it simple somehow makes their work sound less sophisticated, but in reality, the root cause is always something simple. A small handling issue, a temperature fluctuation, or a missed wash. And it's like, I would say, when your mobile doesn't charge, before you blame the hardware or software, you check the cable, right? And the same logic applies in the molecular work. Complexity rarely saves you, but simplicity always does. And then I have seen this many times that once people are in the lab, it's become like a race. Who can finish first, who can tick the box fastest, but don't just copy someone's hand. I mean, learn the deep rationale behind every step and reagent, because I have watched people, for example, copying my protocol, run fast, feel proud that their first attempt works. But the problem is when the condition change, low input or a stubborn sample, then they get stuck. They are stuck. They go in circles because they never learned why the protocol works. So to me, the real expertise is knowing what to change and why, not how fast you can execute a recipe. And finally, before you start, make sure you've got everything ready within your reach, every consumable reagent, because even a tiny delay at a wrong moment can damage the outcome in a meaningful way. I always say, half of a successful experiment happens before you start pipetting. And make sure that you have all the instruments you need to book in advance, because you don't want to end up waiting for in the middle of a critical step for a thermal cycle or a centrifugation, right? So that's why my second rule after patience is timing. It's like a king of everything to me. Think of it as your invisible reagent. You can see it, but it's present in, believe me, it presents in every single reaction and silently deciding whether your data shine or fail. So yeah, at the end, the true expertise, if you ask me, I would say, isn't about complexity, it's the simplicity. And the science isn't about the making things complicated, it's about understanding them to make them look simple again. So having those basics in your mind, just a few, I know that I think I'm running out of time, just a very few points about the transposition. I'm going to be fast. So you need to be extra gentle pipetting the reagents after adding transposition, and no centrifugation or vortexing after adding transposition. And nuclei counting is super important here, because we want a fixed ratio of the enzyme to cells, whatever is recommended in the protocol, because we don't want to end up over tagmentation or under tagmentation, which happens if you don't use the right nuclei concentration. And then again, because of the time, please feel free to take a screenshot and then ask me question if you have anything from this slide. Avoid bubbles, and actually, let me tell you one of these. Yes, avoid bubbles, and I mean in every step, not just during gem generation. To me, bubbles are like silent troublemaker that easily go unnoticed, that can affect the consistency. And another thing is the cross-contamination. I always make a rule to leave an empty well between two samples. Another thing that I am really strict about is that I never ever use the same pipette tip twice. I have seen people, they are dispensing reagent into a series of tube, and because they assume that they are not dipping or immersing their tip into the content of the tube, there is no chance of contamination. But believe me, how would you know that there isn't a micro splash, from the tube that you are putting your tip into that? So anyway, yeah, please feel free, take a screenshot and ask me a question if you had later. And then moving to the sequencing metrics, I have summarized some of our web summary of my 13 multiome GEX, additional 16 GEM-X. We have added these GEM-X to boost our statistical power. So you can appreciate that across all of these metrics, that my all library are remarkably consistent, even though they were processed in different batches. And that shows how excellent and clean performance across the board. And the same goes to GEM-X. Sorry, because of the time, I'm gonna go to the next slide, and then projecting those metrics on the UMAP, you can see that on the left is an integrated all cell from Multiome GEX, and then on the right is the whole cell from GEM-X. And despite coming from independent assay, the same cell type clusters appear in both, which reflect quality and reproducibility of my data. And here I'm showing you that all major cell type clusters are represented in every patient. And then moving to attack metrics, you can see there is, again, a strong consistency across all of my 13 library, which shows that the attack side also performed with high reproducibility. Only point here is that because the sequencing saturation was lower, which is normal, because in attack we are dealing with about 200,000 features. So we decided to resequence to achieve higher sequencing saturation. And as you can see, it resulted in twice more sequencing saturation. And another important QC metrics in attack, which reflect your nuclei prep, is nucleosome periodicity or fragment size distribution. So you know that the transposase insert adapter into accessible DNA, right? So most efficiently into nucleosome-free region, like here, but it can also access the linker DNA. So in this schematic, TN5, you would see that the shorter fragments arise from open region, while longer are from nucleosome bound. So as a result, the size of the DNA fragments reflects how the chromatin was organized. And in a high quality attack data, we expect an enrichment, as you can see, across all of my library. We expect enrichment around nucleosome free, compared to fragments that coming from mono or multi-nucleosomal. And then when you plot these fragments, you see that you would see this characteristic periodic pattern. And this nucleosome periodicity is a hallmark of well-preserved nuclei, meaning that the prep was done in a proper and clean way. And this is, again, our 13 multiome attack. And I think it's the last one. So another attack metric that reflects your nuclei prep is TSS score. That shows how fragments are distributed around transcription site. And then ideally, it should be enriched around the TSS region and gradually decrease to upstream and downstream. And now you can appreciate here across all of my 13 multiome attack, we are seeing a consistent high enrichment just around the TSS. This is the last one. So now that we saw RNA and attack quality metrics, let's look at the relationship of these two modalities. This joint QC compares the total N count in RNA and attack. In a good multiome data, you should see a positive diagonal trend from bottom left to top right, meaning both assays perform well in the same nuclei, indicating that consistency between RNA and attack modalities, meaning that nuclei with high quality RNA also have rich attack profile. And lastly, just showing you that this is the integrated data from attack. Clustering here works based on cells that share the common open regions, same pattern. And now you can see that our integrated attack is largely concordant with our RNA data, showing a strong consistency between these two modalities. And yeah, in a summary, I showed you a reproducible nuclei prep that resulted in a clean and consistent attack signal, along with a strong concordance of RNA and attack modalities. And I just want to highlight that while I have done the all wet lab, the large scale data analysis has been is led by our highly experienced bioinformatician, Andy, together with his RA Felicia. And I just want to highlight that Quay and Kat from 10X, they have been incredibly supportive, honestly, without any exaggeration. And they're super helpful throughout the entire, not only this project, but also my Flex on rare breast cancer. And also my colleague, Michael from our lab. He's been truly like a living encyclopedia always for me. And also, I want to appreciate everyone who helped me to make this work possible. First and foremost, my incredible and kind supervisor, Jane and Jeff. Actually, Jane has been truly exceptional, always inspiring, full of ideas, and deeply supportive. And trust me, her enthusiasm creates such a positive environment that it's impossible not to feel motivated. And honestly, none of this would have been possible without her constant support and encouragement. And over the time that I had the privilege of working with Jane, every day her energy and vision have made each day as exciting and rewarding as the very first day for me. And that's why he's a big source of inspiration, which shapes my passion for diving into breast cancer more and more every individual day. And I've been very fortunate to be surrounded by all of these beautiful and supportive people within our lab and beyond. And I have to confess that all the labs and colleagues I have acknowledged here, they genuinely feel like my family, and working with them has always been an absolute joy. So with that, I leave it with you, Martha.
Wow, Somi, thank you so much for this presentation. It was really amazing. And I just jotted a few things that I liked the most. I love the timer trick for different samples, that how you just put several timers. How many timers do you have in the lab?
Thanks to companies like you that at each conference we try to.
Okay, that's noted. We will give away more timers. Okay. I love the Somi principles of success. I think it's just so truthful. Like, even if it wouldn't be to 10X experiment, but to any other lab work, or probably even not in the lab. So that was fantastic. And especially the why behind the methods that you are learning, not only how, but actually you really need to understand what's happening in the reaction to actually be able to perform it like at its full capacity. We have a question online from Hamid. Thanks, Somi, for your comprehensive talk. I wanted to ask how the multiomics approach between Xenium and single-cell RNA-seq differs from when you're using Visium HD 3' and single-cell RNA-seq. I think Jeff can also help answer this question.
Sorry, what was the question, Marta?
So basically, how working with single-cell assays and Xenium for you was different than if someone would be working with Visium HD 3' and single-cell assays?
I mean, the concept is the same, but it mainly depends on the nature of the samples, your sample archive. If it is fixed, so yes, you have to use FFPE, but still you can use, I mean, I'm not sure about the Visium 3'. That needs fresh sample. Am I right, Jeff?
Yeah. Yeah.
So, yeah. I mean, I can tell you that it depends on the nature of your sample. If you are working like in our rare breast cancer project with fixed sample, so you have to use, I mean, you have to use, FFPE, Flex, and then you have either can choose Xenium or Visium, not the 3'. But then if you have fresh samples or frozen samples, you can choose any other 3' assays like the GEM-X or Multiome and use the 3' Visium HD. But Jeff, you please.
Yeah. Basically, if you have fresh frozen sample, you can use any 10x assay, including Flex, because you can still use Flex in that situation. Yeah. But if you have fixed samples, so FFPE, you are a bit limited in the numbers of assay you can use. I don't know, Hamid, if it answers your questions, and if anyone has any more questions. Jeff, do you have any questions for Somi?
I just want to say thank you so much, Somi. I really appreciate you, I guess, sharing your knowledge and tips to the broader community. I think it really does make a difference. All these little bits of like what might seem like minutiae are really, really important. You know, the pipette tip stuff and the timers and all these little things really do add up to make a very successful experiment. As Marta highlighted at the start, Multiome can be a tricky product to work with because the sample prep is extra critical, even more critical than sample prep normally is. So the more thorough you can be there, the more guidelines you can follow, the more practice you can do, the better your outcome. So I know that wasn't really a question, but I do want to say thank you, Somi, for sharing such great insights today.
No, all right. Thank you again, Somi. We don't have any more questions. So thank you, Somi. Thank you, Jeff. Thank you, attendees. And stay tuned for the next episodes of Cellular Conversations.
Thanks, Marta. Thanks, Jeff. Thanks,
Thanks,
everyone.
Somi.
Bye. Have a good day. Bye.